what's in the periodic table?
Okay.
It's the same as the, oh, sorry.
Okay,
I know you know.
the number above the like top element is.
Yes, that's perfect.
I have a question about the BMC conference. How many days and what days?
All right, let's just go over a few more things on here first, and then we'll move into the BMC conference. So. I'm moving on to. So who remembers what the kind of weak force between water is? Steven.
Hydrogen bond?
Cool. And what's kind of the importance of hydrogen bonding between water? Anyone know? Yeah.
It has a 105-degree angle, so it's slightly polar.
Yeah, that's really good. The important part is that it means that water is a polar molecule, and it means that it kind of allows different things to happen in our bodies. It allows the kind of difference between cell membranes, which we're going to look at one more time, which are non-polar on the inside and polar on the outside. Really just kind of allows water to, stick to you, and it allows for that kind of like viscosity that it has. Yeah, the fluidity that it has. It allows for our blood to be circulating. It allows for the state that our brain is in. So who remembers why we're not made of silicon? Yeah.
It's not readily available.
That's good on. There's another reason too. Marlena.
It's too big.
Yeah. And how many valence electrons does silicon have going around its outer shell? 4. So, I don't know if we went over this, but who knows how many bonds nitrogen likes to make? It's kind of a trick question.
So it's in column 5A and it needs to have like some.
Right. So Zane, why does it like to make 3 bonds?
Because the second shell is 8 electrons, and then it already has 5 in the second shell.
That's good.
And it needs 3 more.
That's good. But so if we think about it, so if it's making 3 bonds, it always wants to have 8. So if it's making 3 bonds, that means there's a lone pair of electrons on it. So we have 2 bonds, 3 more bonds, that's a total of 8. Each bond counts for 2. So 2, 4, 6, and then 2 more electrons, that makes 8. Alright, trying to get to the So who remembers what the 4 biomolecules are? Yeah.
Is it carbon, nitrogen, oxygen, and phosphorus?
Okay, those are the actual elements. But the difference between it's okay. The difference between a molecule and an element is that a molecule is made of different elements. So like when we talk about something like dopamine right here, we can see that there's a carbon here and an oxygen here and different things. This is a molecule. So when we talk about different types of biomolecules, we're thinking of 4 classes. Yeah.
Carbohydrates, lipids, proteins, and nucleic acids. Yeah, I totally remember that.
Cool. So what are proteins made out of?
Amino acids.
Awesome. And how many amino acids are there?
Hella.
Hella? What's inside? Yeah, there's 20 amino acids. There they are. So pretty. And what's interesting is that a lot of the amino acids are precursors to different neurotransmitters. What's the one you get from turkey?
Tryptophan.
Tryptophan is a precursor to serotonin. So when one of the reasons why, like when you eat after Thanksgiving dinner, you're really stuffed and you're like, oh, I feel kind of sleepy and like I feel like really good. Well, that's because you just got a bunch of tryptophan, and it's actually a precursor to serotonin, which is what we're going to talk about later in the neuroscience lecture. Yeah, Alex.
That part's not really true.
Yeah, I was gonna say,
Turkey does have tryptophan, but most other meats and like chocolate and like a bunch of other stuff has way more tryptophan than turkey, and it really is just kosher super stuff.
yeah,
And when it's super stuff because it's higher. But it does have tryptophan, and I believe whatever you're going to say about tryptophan.
Okay. Well, regardless of how much tryptophan there is in turkey, I will tell you that I'm 100% sure that tryptophan is a precursor to serotonin. But thank you for my correction.
I want to make a little comment about this.
Yeah.
So, like, why do you think the protein forms into that, like, three-dimensional structure versus, like, another one?
Is it so it can like make the binding sites for the
Like, yeah, that's, like, its function is related to it, but why do you think it forms one structure versus, like, another structure?
And it has to do with like the polarity on the molecule, and also with like how like what kind of bonds it has on it too.
Yeah. So in physics, it's called like the free energy, and like the protein's always trying to minimize the free energy. It's always trying to go to the lowest energy state. So all those bonds are trying to minimize in a geometry, which is like least resistance. And then like the geometry of the protein like is like really, really correlated to its function. So I think that's pretty interesting.
So what would be like the most basic then? Because there's like 20 different types, like does it just get more and more basic until it has like the laziest or most relaxed state, right? I don't know. I think
So-
you need like a 3D diagram. to see that, right?
I think I could shed some light into that. You know, what's really interesting when we talk about something like DNA replication, which we didn't really get into hugely yesterday, our body uses a template strand of DNA. So it's like there's this template strand of DNA that we have to replicate. That's kind of how we thought proteins folded. That's what biochemists thought back in the day. They're like, well, we have this strand of somewhere between 10 and 100 amino acids, and there must be some sort of template for how these proteins are folded. In fact, there's nothing. It is literally what David said, the lowest energy state for which these proteins fold. And if we look at the amino acid structure, the exact sequence of the amino acids, biochemists cannot figure out how it's going to fold until they actually watch it fold. And then they watch it fold, and it folds into the same configuration every time. So it's kind of crazy.
It's really crazy that it would actually form in the same configuration each time.
Yeah.
if it does hold the same way each time, would that mean that it has a designated route that it goes, and you could formulate why it does that?
Yeah, no, you definitely could. No, theoretically, it's possible. We literally, when I was listening to Michael Stone, who's my biochemistry teacher, he's like, we literally do not have a supercomputer that's powerful enough to figure out all the possible combinations of all the different polarities on these different atoms, that the way it's going to fold. We can't figure it out. Whereas something like DNA, we can look at it and be like, well, it's going to be synthesized this way because we see this template right here. Okay. I think just going over the phospholipid membrane is just a really, really important structure because this is what keeps water out of the cell. The reason why water is being kept out of the cell is because of the same reason why that oil that I showed you guys last time was staying on top of the layer of water. This inside here is made of this oily substance which is not allowing water to come into the cell. And it's not allowing other kind of big polar molecules to come into the cell without some sort of transport. It's going to come in. So a lot when we go into neuroscience, we're going to be seeing lots of proteins that are actually embedded into the membrane. And that's going to allow different ions that are charged, like sodium and potassium, to come in and out of the cell, which is going to be responsible for how neurons fire, how our brain works. So this is kind of just what we're setting up.
Important for regulation, the membrane.
But what's the opposite? Or go back real quick. Like, what's the plant version of cellular respiration? Photosynthesis. Yeah. So, can I use this opposite? But,
when you have like, you know, a little plant.
We have a little thing on how DNA was discovered. Pretty cool. I think we'll take a look real quick in the molecular biology lecture. I don't want to go in depth in the molecular biology lecture, but we'll look at how DNA was discovered briefly. It's a pretty cool story. Okay, cool. So who can recite that example that I gave you guys right at the end of class, like really fast? I feel like everyone who has taken a science class was like, yeah, that was a really good analogy. But for those of you who maybe haven't taken a science class, that was like a little